Picture this, we're zooming deep inside a living cell. We fly right past the famous nucleus, past the powerhouse mitochondria, and dive into the bustling, fluid-filled city that is the cytoplasm. It's right here in all this organized chaos that we find some of the cell's most amazing and absolutely vital molecular machines. Today, we're getting to know two of them, the mighty proteasome and the mysterious vault. So we all know the big names in the cellular world, right? But what about the unsung heroes? You know, the intricate little machines working tirelessly in the background, keeping everything in order, transporting goods, and cleaning up all the messes? Well, that's exactly what we're going to get into. All right, here's the game plan. First, we'll see what makes these organelles so different. Then, we'll meet the proteasome, the cell's personal recycling plant. After that, we're diving into the secrets of the vault particle, which is kind of like a molecular transport capsule. We'll put them side by side, and then we'll look at why they are so incredibly important for the future of medicine.
So, what really sets these guys apart from something like a mitochondrion? Well, the answer is pretty simple, actually. They are rebels without a membrane.
That's right. Unlike most of the organelles you probably learned about first, proteasomes and vaults don't have that fatty lipid layer wrapped around them. Nope, they are pure protein-based machines. Well, in the case of vaults, it's a ribonucleoprotein, which is just a fancy way of saying it's a complex of both RNA and protein. They just float freely all through the cytosol, and that freedom lets them get right where they need to be, handling these super important jobs like protein quality control, transport, and cell signaling. Okay, let's start with the proteasome. Think about it. Every single factory needs a quality control department and a recycling center, right? Well, for the cell, the proteasome is both. It is an absolutely essential piece of machinery for keeping the cell healthy, clean, and running smoothly.
So what is it exactly? A proteasome is this hollow, barrel-shaped protein complex, and its only job, is to find and destroy other proteins that are either damaged, folded wrong, or just aren't needed anymore. The protein shredder analogy? It's perfect. It literally takes in unwanted proteins and just chops them up into reusable little bits.
Now take a look at this. You can really appreciate the beautiful architecture of the proteasome here. You've got that barrel-like structure in the middle, that's called the 20S core particle. That's the shredder part. The destruction happens inside its central chamber. And then at either end, you have these caps, the 19S regulatory particles. Think of them as the bouncers at the door, deciding who gets in and prepping them for disposal. And the whole process is just incredibly efficient, like a molecular assembly line running in reverse. First, a protein that's marked for destruction gets tagged with a tiny molecule called ubiquitin. It's basically a cellular sticky note that says, "Trash me." The 19S cap sees that tag, grabs the protein, and uses a burst of energy to unfold it from its complex shape. Then, it's fed right into that central chamber where all the peptide bonds holding it together are sliced apart. The end result? Tiny little fragments and amino acids, which are released back into the cell to build brand new proteins. See? Nothing goes to waste.
Now, this is a really crucial point. This isn't just simple housekeeping. This is fundamental for survival. By clearing out all that junk protein, proteasomes prevent the kind of toxic buildup that we see in devastating neurodegenerative diseases like Alzheimer's and Parkinson's. They are the guardians of what scientists call proteostasis, basically keeping the cell's entire protein environment in perfect balance. When that balance gets thrown off, things can go wrong fast.
Okay, so we've seen the cell's super efficient recycling plant. Now let's turn to something a lot more mysterious, the vault. If the proteasome is a recycling plant, the vault is like a high-tech, multi-purpose transport vehicle, and we're still figuring out everything it can do. And you know what? Its discovery was a total accident. Yeah, seriously. Back in 1986, a scientist named Dr. Leonard Rome and his team were studying something completely different, but they kept finding these weird, barrel-shaped things contaminating their samples. Turns out they had stumbled upon something nobody had ever seen before, a massive particle made of both protein and RNA. It was, and still is, the largest ribonucleoprotein ever found in cells like ours.
And this is where it gets its cool name. When they finally got a good look at its structure, with all those intricate, repeating arches, it immediately reminded them of the vaulted ceilings you'd see in a grand cathedral. It's a really fitting name for such an elegant molecular structure. You can totally see the resemblance here, can't you?
Now, for something so huge and complex looking, the vault's recipe is surprisingly simple. Check this out. A whopping 75% of its entire mass comes from just one type of protein, And so, fittingly, they called it the major vault protein, or MVP.
So you've got the MVP forming that main barrel shape, and then tucked inside are a couple of other minor proteins, VPARP and TEP1, and this one small, really intriguing strand of RNA called vault RNA, or vRNA. And honestly, the exact job of this little piece of RNA is still one of the biggest puzzles about the vault.
Which, of course, brings us to the big question. What does this thing actually do? Well, the answer seems to be a lot. Research has tied vaults to a whole bunch of critical jobs inside the cell.
Scientists think vaults act like tiny cargo trucks, hauling molecules between the nucleus and the cytoplasm. They might also help place messenger RNA in specific spots where it's needed, or act as a kind of scaffold for communication pathways. They might even help build the doorways into the nucleus itself. But one of their most studied roles, well, it has huge implications for medicine.
And this is the critical clinical link. In a lot of cancer cells that have learned to survive chemotherapy, scientists find way more vaults than usual. The leading theory is that these vaults can actually grab the chemotherapy drugs, package them up, and ship them right out of the cell, protecting the tumor. This makes the vault a huge target for researchers trying to develop better cancer treatments.
All right, so now that we've met our two cytosolic machines, let's put them side by side. This will really help us lock in their differences and appreciate just how distinct their jobs are inside the cell.
This table lays it all out pretty clearly. On one side, you have the proteasome. It's a pure protein degradation machine. Its whole purpose is to break things down. On the other side, you have the vault, a ribonucleoprotein transport complex. Its job is to move things around and regulate processes. One is all about quality control and recycling. The other is about transport and regulation. Totally different structures, totally different jobs, but both are absolutely vital.
So why does any of this matter outside of a biology class? Well, because understanding these incredible little machines is opening up a whole new world of possibilities for medicine and technology.
So here's the main takeaway. Proteasomes are the absolute masters of quality control. Learning how to work with them is key to fighting diseases caused by protein buildup. But vaults could be the future of nanomedicine. Because they're basically natural, non-toxic hollow capsules, scientists are incredibly excited about their potential.
And this is a glimpse of that future. Researchers right now are actively engineering these vault particles to act as tiny, programmable nanocapsules. Just imagine loading them up with cancer drugs that they deliver only to tumor cells, or using them to carry vaccines directly to your immune system. It is a revolutionary idea, a whole new platform for medicine, and it all started with someone noticing a weird contaminant in a test tube. It just leaves you with this amazing thought. These tiny machines have been running inside our cells for millions of years, and we are just now beginning to understand them, to speak their language, and even to redesign them for our own purposes. So as we get better at engineering them, you have to wonder, what currently untreated diseases might we finally be able to conquer? The possibilities are truly staggering.